Okay, so I just wanted to clear up a few items that you need to know for your test. The first thing that you need to know is the difference between a plant and an animal cell. Don't forget there's only a few differences. The plant cell has a cell wall, which is this really thick part outside of the cell membrane. Don't forget both cells have a cell membrane, but the plant cell also has a cell wall. Chloroplast is something else that a plant cell has that an animal cell does not, and that's where photosynthesis takes place. And then plant cell also has a huge vacuole. Look at the size of the vacuole here. It's and you guys know that that's for storage of water. So if it doesn't rain for a long time, the plant still has a source of water. The animal cell, on the other hand, there's only a couple of differences. I'm going to focus on the centriole, and the centriole is this. It sort of looks like two pieces of pasta side by side in an animal cell, and that's where the spindle fibers, it makes the little centrioles to pull the cell apart during mitosis. So the centriole is something that's present in an animal cell and not in a plant cell. So I've put that in an easy chart for you. So you see that there's a large vacuole, cell wall, and chloroplast in a plant cell, and an animal cell has centrioles. And all the other stuff that we need to know about are present in both. Here's another chart for you to make it easy. These are very typically tested organelles. Make sure you know what the mitochondrion does, the endoplasmic reticulum, what it does, and don't forget there's a smooth and a rough, and the smooth has to do with proteins, and the rough has to do with, the smooth has to do with lipids, and the rough has to do with proteins. Ribosome, very important for protein synthesis. Don't forget chloroplast is the site of photosynthesis, only found in a plant cell. The Golgi, this is the one that's really cool at transport things, so the cell puts things in it, like, I don't know, proteins and lipids, and it gets processed and then packaged in a little vesicle. And sometimes the little vesicle will fuse with the cell membrane. I'm pretty sure you guys remember the vesicle comes up and actually becomes part of the cell membrane, and then it expels its contents out. So that's exocytosis. And don't forget, we have to know the difference between endocytosis and exocytosis, and endocytosis is just the opposite. So if there's something outside of the cell, the membrane actually sort of pinches in so that the stuff can go in, and then it makes a vesicle like this, and then the vesicle comes into the cell. Okay, so, and that's, you know, transport of large contents. The next thing that you have to understand is the cytoskeleton, and these are, this is the cytoskeleton. It's kind of weird that a cell, you know, that would, tiny little thing would have a skeleton, but, and they're made up of microtubules. So tubules, long tube, micro means tiny. So tiny tubules make up the cytoskeleton, and that helps to give the cell some structure, just like a skeleton in our bodies give us some structure. And then the last thing that I want you to see is, typically on tests they'll give you diagrams of animal and plant cells, and typically the plant cells are sort of rectangular shaped. And if you look here, huge vacuoles, look at this as a plant cell, giant vacuoles. There's the nucleus and the cytoplasm, and all of these structures, look at how many there are. In a plant cell is going to be chloroplast, because a plant cell, that's its purpose, is to make food for the plant, so there's going to be a lot of chloroplast. So there's just a few things that you have to remember.